Hello and welcome everyone to APFED's EOS Support webinar series. My name is Jennifer Roeder. I'm a communications consultant for APFED, and I'll be moderating today's webinar about hypereosinophilic syndromes and your heart. Joining us today is Dr. Nives Zimmerman. She is a physician scientist at the University of Cincinnati in Ohio. Clinically, she is a pathologist. Her research focuses on eosinophil and mast cell mediated diseases. Welcome, Dr. Zimmerman.
Thank you very much. Uh, I really appreciate, Jennifer, the introduction, and I appreciate the invitation to speak today with you. I will take a second to find my slides. Okay, so hello, everybody. Again, I'm really happy to be here to talk to you today about some of the work that my research group has done, specifically focusing on hypereosinophilic syndrome and the heart. But I first wanted to put a little background about myself and our work, and then the topic in general. So I'm very passionate about rare diseases and hence my zebra, but it's as you all know, rare diseases are defined as ones that affect fewer than 200,000 people in the United States. And if you add them all up, that's about 7,000 diseases, which really end up affecting about 25 to 30 million Americans. And so it's a sizable number of people. And while, of course, cancer is important and diabetes is important and et cetera, I really think that research in rare diseases is critically important. And as many of you know, unfortunately, because there is not as much research and work in these areas and for many other reasons, many patients suffer from missed diagnosis or delayed diagnoses. And then even once we get to, okay, this is what you have, it's very often that we don't have treatment options. And so for those reasons, I'm really passionate about trying to change the outcome for patients with rare diseases in general, but I'm in particular interested in eosinophils and mast cells. The other important thing, and Jen has mentioned, is that I am a pathologist. So I don't directly manage patients, but my emphasis is on diagnosing disease. And my specialty is hematopathology, meaning I am mostly involved in blood and bone marrow diseases, such as neoplastic hypereosinophilic syndrome. And so this picture here is one from one of my patients where you see the bone marrow, which is diffusely involved by eosinophils. And I can tell it's a bone marrow because I think, let me make sure I can have my, well, sorry, I'm trying to get the marker. I forgot to get that ready. No, that's not what I wanted. Okay. There we go. Okay. So we know this is a bone marrow because there are here these two really big cells that are different than everyone else. Those are megakaryocytes. And they are the only thing that's in this picture that should be in the bone marrow. This whole infiltration of eosinophils is abnormal. And this is a very abnormal bone marrow involved by way too many eosinophils. And so as a pathologist, I look under the microscope, but I also integrate information from many other modalities, such as molecular testing, cytogenetic testing, etc., to come up with a diagnosis for a patient. Um, and when I look under the microscope, the what I think of it is as being a detective, like, oh, what happened here? Why is this bone marrow so abnormal? For instance, in this case, what happened? It's kind of like coming to a crime scene and trying to figure out from one snapshot what actually is going on. And that has always been what I was passionate and curious about, and has also driven my clinical work, but also my research. So I've been doing research in eosinophils for almost 30 years now. And over those 30 years, I've asked basically two main questions. Why are they here? Why is there so many eosinophils? And for a while, I was looking at asthma, so we were looking in the lung. I look in the GI tract, bone marrow, etc. But the overarching question is, why are there so many eosinophils here, and how are they causing damage? And so that's kind of the mechanistic stuff that has always driven the research side of sort of my brain and my work. So I'll take a step back and just talk before we get into eosinophils and the heart, just take a general overview. As many of you know, there's a spectrum of eosinophil-associated diseases, and that includes from eosinophilic asthma, other allergic disorders, parasitic infestation, eosinophilic gastrointestinal diseases, to the more rare ones, the sort of the zebra side of eosinophilic leukemias, such as the one that I've shown you in that bone marrow, hypereosinophilic syndrome, and eosinophilic granulomatosis with polyangiitis or EGPA. Now, the one thing that's interesting to me is that basically, no, irrespective of why a patient has really high eosinophils, these cells have these nasty granules that, if released, can cause damage in the tissue. And so, irrespective of why someone has high eosinophils, they can get tissue damage. And that tissue damage can manifest in skin, in the lung, in the heart, and in many other organs. And that will be different in different patients. So what we will focus on today is mostly the ones that tend to have really high eosinophils, so hypereosinophilia, so hypereosinophilic syndrome and its related diseases. And specifically looking at damage in the heart. So for the last 5 years, I have focused mostly on the heart, and this slide highlights some of the reasons for why that is. So on the left-hand side here is data from a paper where many groups got together to try to put their patients together into a big study, because the problem with rare diseases, as you know, is that most centers will only have a very limited number of patients, and it's really hard to draw general conclusions or learn about them, about a specific disease, if you don't have a high enough numbers. And so this was a fantastic study because multiple centers came together and looked at certain things about hypereosinophilic syndrome in a organized manner, so that all of the centers are looking at it the same way. So for instance, here we're looking at the percent of different organs that are involved in patients at presentation on top, and then any time during disease progression. And what you see is that skin, lung, GI tract are the most commonly affected organs, both at presentation as well as at any point during the disease progression. And then there is a variety of other organs that could be involved. I've put an arrow here for the heart, which is about 5% of people at presentation, about 20% at any point during the disease progression. So this is not necessarily the highest organ that is involved. The other thing that they noted is that when you look at patients with hypereosinophilic syndrome, heart involvement tends to be a little more common in patients with neoplastic hypereosinophilic syndrome, such as those eosinophilic leukemias, than patients with reactive hypereosinophilic syndrome. However, even though it's not as common as skin, lung, and GI involvement, it does cause a lot of morbidity and even mortality. So historically, about 45% of death from hypereosinophilic syndrome occurred due to heart disease. And in a more recent study, it was about 1/3, so about 33% of deaths were because of heart disease. So even though it's rare, it is, in my mind, very important. And as I've mentioned, as a pathologist, the other thing that we do is perform autopsies and look at patients who have died to understand what led to their death. And so the picture on the right is actually slices of a heart from one of the autopsies I performed when I was a resident and learning how to be a pathologist, which was from a young lady who knew that she had hypereosinophilia, but we just didn't have the time to really figure out what kind and really understand it better and treat it better. And she unfortunately died. And these are slices of her heart. And what you can see normally, this should be a hole, but there is a thrombus here. And you can also see it on this last slice as well, where there were multiple thrombi in her heart that really made the heart not be able to function, not be able to send blood throughout the body. And she unfortunately experienced sudden death. So, and this highlights some of the reasons why I'm really passionate about trying to understand why do some patients develop heart disease in hypereosinophilia, and what are the mechanisms of the heart disease in patients with hypereosinophilia. So what are some of the remaining gaps in knowledge and some of the barriers that we have in disease management? One of the big ones is early diagnosis. So many patients with HES or other reasons for hypereosinophilia have this asymptomatic stage where they're not having any symptoms. They're not having chest pains. They're not having any symptoms. But their heart, if you were to look at it, is actually involved. And so we're really lacking sensitive and specific biomarkers for this early diagnosis. And I will talk a little bit at the end of some of the things that we're doing to try to improve the situation here. The other thing that's a problem in clinical management is that there's lack of disease-specific consensus guidelines for treatment and management. So while we've made a lot of progress in treating HES, and that does help with the heart disease aspect of it as well, it's still not perfect. You've seen that there's still patients who develop heart disease, and there are still patients who die from it. And so there are some nuances that we need to do a better job understanding where, in addition to treating the hypereosinophilia, what are the specific heart things that we need to do to improve the outcomes for patients? And I think for both of the above, understanding the disease pathogenesis is really important. Um, and, so understanding uh, why do um, some patients get heart disease and others don't? Why do some patients have super high eosinophils in their peripheral blood and they don't even have any organ involvement? So those are all some really important questions that um, many labs and groups are thinking about and working on. So I will start with understanding of disease pathogenesis because that is where my lab has done most of the work, and we've had a recent publication, which is how I got in touch with APFED and Jen to um, come and talk to you today. So starting about what do we know about disease pathogenesis in terms of heart involvement in um, hypereosinophilic syndrome? So there's really not much known in human disease, and most of what we know is from a series of autopsy studies that were conducted back in the 80s and 90s, which were really helpful because we get a feeling for the progression of the disease. So it seems to start with the endocardium, which is the inside of the heart. So the layer between the blood that the heart is pumping and the actual muscle. That's where it's initially thought that the first damage occurs. Then it involves the muscle or the myocardium, which gets infiltrated by eosinophils. And then that leads to a lot of these muscle cells dying. And then as the muscle cells are dying, they get replaced by a fibrosis. And then the endocardial lining, because there's damage, that's where the thrombi occurs, such as in the patient that I've shown you. So that's what we understood from the human studies. And what I'm showing you here is a picture of a biopsy from a, uh, a paper that was published by Dr. Gleich and his group in the New England Journal of Medicine, where they actually performed a biopsy, which is not routinely done, but in this particular situation, it was done. And so this is just a small piece of the heart muscle. And you'll have to believe me, but that this is the endocardium, so that inner layer. And actually, this muscle, at first glance, I don't see any eosinophils. And even here on a higher power, these are your typical muscle cells, these pink cells with the blue nuclei. This is the endocardial layer, so this would be the lumen of the heart. I don't see any eosinophils. But when they went ahead and stained with an antibody to MBP, it really lit up. And so this bright green is the immunofluorescent staining for anti-MBP. And especially in this endocardial part, you can see that that's very strong. And the thought here is that when the eosinophils degranulate, they're sometimes completely unrecognizable as eosinophils. So they don't look as beautiful as that one picture I've shown you in the bone marrow, but their granule proteins have been laid over here, and then they can cause damage in those cells. So that's sort of what, as much as we could know from human studies about at least the natural history of the disease progression. But in order to be able to better understand the mechanisms of disease, in our lab, we have developed a mouse model. This is our second mouse model and I won't go into details about the first mouse model that we used because this new one has been really helpful and will be the focus of the studies that I will show you today. So how does this work? In this particular model, which we call eosinophilic experimental autoimmune myocarditis, which is a mouthful, but I'll call it EOEAM, and I'll explain to you what happens here. So basically, we take mice, and these are hypereosinophilic mice. So they're IL-5 transgenic. These mice have high levels of interleukin-5 that is produced by their T cells. That's where the transgenic part is. That's the genetic manipulation that we have done to these mice. So because they make a lot of IL-5, their bone marrow makes a lot of eosinophils, and they have a lot of eosinophils circulating. But like those patients with benign eosinophilia who have a lot of eosinophils at baseline, these mice tend to not have a lot of problems. So at baseline, I wouldn't say they have zero problems, but they are fairly close to normal. And so what we do in this scenario is we challenge them with a peptide from a heart protein that is in an adjuvant. Basically, we're tricking the immune system to not consider that heart protein as self, but now it considers it as a foreign thing that it needs to attack. So the immune system mounts a very strong response against this heart protein and then attacks its own heart. And that's what I will show you the pictures on the next several slides. But basically, we do this injection twice, and then we characterize the mice mostly at the 3-week time point. I will show you some pictures from the 6 weeks time point as well. We have several controls. So the one that causes the myocarditis or the heart damage will be this treatment peptide that most of my slides will have labeled as T. And then sometimes we have a control with a control peptide or a unchallenged control. So what happens to these mice? Okay, so these are pictures of slices of a mouse heart. The one on the left is a normal mouse heart or that has been challenged with this control peptide, but nothing's happening here. It looks pink. This is the wall of the heart. This is the inside lumen. I don't have the entire 4-chamber cut, but believe me that this is the septum in between the two main chambers of the heart. So this is the lumen, muscle, it's pink, it's normal, nothing bad's going on here. But then the one that received that peptide from the heart and we tricked the immune system, what you see all of a sudden is a lot of this blue stuff. That is your immune cells that are coming in and attacking this heart. And when we look on high power, there's a mixture of cells. There are lymphocytes, there are eosinophils, there are macrophages, there's a mixture of cells. And the way I know that here shown again is immunostaining for major basic proteins. So this will label your eosinophils. And you can see that there are plenty of eosinophils in this heart. And then I love this picture because it really kind of shows what's going on. So down here is a relatively normal muscle cell. It's pink, intact, has a nucleus. But then look at these muscle cells. They are all very frayed and kind of falling apart. And then on top of them, there are these inflammatory cells, including eosinophils, which have these very red granules. And it looks like this eosinophil has kind of fallen apart, and these granules and probably the granule proteins are attacking and fraying this muscle cell. And so I think this really shows what we think is going on as a in a sort of morphologic way, that eosinophils are causing damage to these muscle cells. I mentioned in the humans that once the peak of inflammation subsides, there is fibrosis. And we looked at that in mice as well. This is at the later time point and using a different stain. So trichrome stain in normal mice, which is what you see on the left, stains the muscle very red. And that looks perfect. So normally in the muscle, it's mostly muscle cells with just very little space in between, because all of those muscle cells are working together to pump the blood through our body. But the one on the right is showing a mouse that was challenged with the myocarditic peptide, so the treatment peptide. And what you see is that these red cells are separated from each other, and that now we have a lot of blue, which is collagen, which is fibrosis. So altogether, what we show is that this mouse model shows similar histologic features to human disease, which include myocardial infiltration by inflammatory cells, including eosinophils. It includes damage to the muscle cells, the cardiac muscle cells, and that this is followed by fibrosis. And then while you can imagine that this heart wouldn't necessarily be working well and that there is damage by eye, we've also done some testing. So shown here is looking in the blood, in the peripheral blood, for evidence of damage. Shown on the left is circulating cell-free DNA. And this will become important as we talk about biomarkers and how can we tell whether there is tissue damage. And what you can see is that mice that received the peptide that causes damage do have an increase in cell-free DNA over time. This is at 1, 2, and 3 weeks, while the control ones do not have a significant change in cell-free DNA. Now, cell-free DNA, and I'll talk about that more a little later, is basically when a cell dies, it extrudes its content, including its DNA, and this DNA is now circulating in peripheral blood. So with this assay alone, we just know that somewhere some cell is dying, but I can't tell you exactly which one it is. And so in this situation, we used serum troponin as a measurement of specifically heart damage. So when troponin is a protein that there is a lot of in heart muscle cells. And again, when the heart muscle cells die, troponin gets released and it can now be measured in peripheral blood. And that's what we do for heart attacks. And that's actually what's currently used as a measure in patients with hypereosinophilic syndrome. Do they have heart disease? So you can see that in this mouse model, we also have a release of serum troponin. So together, these confirm that there is heart tissue damage in this model of eosinophilic myocarditis. Okay, so now we have a model, and now our first question was, are eosinophils important? Now, I've shown you that picture of that eosinophil on top of that cell, and I think it's certainly a good suggestion that eosinophils may be the ones that are causing some of the damage. But just because they're there doesn't mean that they've done it. So we went ahead and actually tested the role of eosinophils in the model. In order to do that, we used a mouse model called the delta double gata mice. That's a eosinophil deficient strain of mice. So these mice don't make any eosinophils, even in the bone marrow. And so they're really eosinophil deficient. And this is actually one of an MBP stain from this delta double gata mouse. And I've shown you a few slides back what MBP looks like in a mouse that has inflammation. But in the delta double gata, there are no eosinophils. And then we quantified that over here, and you can see that indeed the delta double gata have no eosinophils, while the L5 transgenic that are challenged do have eosinophils. And then we looked at, well, is there inflammation? And we'll start here with the quantitative data, which shows you that the L5 transgenics, they have different severity of disease. And in the delta double gata, well, there are a few mice that did have some inflammation. The majority did not. So there was a significant decrease in inflammation. And I'm showing you here, this is the worst, like this mouse up here, right? So this mouse did still have some inflammation, but there were no eosinophils. And it was mostly lymphocytes that were inflamed, that were infiltrating the heart in this mouse. And we were unable to see any significant in this particular mouse. While there's infiltration, we did not see that dropout of cardiomyocytes as we did in the ones where eosinophils were present. So from that, we concluded that eosinophils are critical for heart inflammation in hypereosinophilia-associated heart disease. But again, they're not the only ones, and there are other cells that are present there. So in order to start looking at the pathophysiology, we used a relatively novel approach, which is called single-cell RNA sequencing. So the way this is done is that you take the heart and you from the inflamed mice, and you separate all the individual cells, and then each individual cell is assessed for expression of genes in the cell. And that generates a ton of data because we're talking about tens of thousands of cells. And for each of them, we know what level of expression for all of the genes in the genome. So with a lot of help from our bioinformatics colleagues, we could then look at that data and kind of put it back together and see which cells are present using this approach. And you can see that, there is a variety of both structural cells, such as fibroblasts, endothelial cells, as well as immune cells, such as, eosinophils are up here, but we also have T cells, B cells, neutrophils, etc. So there's a lot of different cells, but the beauty of this approach is that we can now tell how different are these cells from each other in the two different groups. Do they interact with each other? And basically, this one experiment has generated data that we are going to mine for several years, probably, to find different pathways and different things to try to better understand how these cells interact with each other. But for today, I just wanted to focus briefly on eosinophils. So here they are, the small population here. For a variety of reasons, eosinophils seem to be very fragile. So even though they are a large proportion of the cells in the heart, in this model, not many of them really survive the processing that goes into this single-cell RNA sequencing. But there was still enough of them from both the control and the myocarditis mice that we were able to ask some questions about them. And so on the right is one such question where we asked, okay, now that we know the genes that are expressed in those cells, so are the eosinophils in the myocarditis mice different than in the ones that are in the heart of a normal mouse? And to do that, we looked at genes that were used as marker genes in another study to look at different eosinophil subtypes. So from that paper, they identified the genes that are markers of eosinophil precursors and immature eosinophils in the bone marrow, then the ones that are seen in circulation, and then the ones that are seen in tissue at baseline and tissue with activation. This was originally done in the GI tract. But interestingly, when we look at the eosinophils from our mouse, what we can see is in the ones with myocarditis, so the ones labeled with T, red being high expression, we see that a lot of the genes that are seen in activated eosinophils are indeed expressed in these eosinophils in the heart of our myocarditic mice. And I'm not going to show you all of the data that we've done, but we've also looked, so even within activated eosinophils, there are several different subtypes, and the particular ones that we are seeing here are the type 1 activated eosinophils. And so we concluded from the data I'm showing you and others that I'm not, that eosinophils in the heart of myocarditis mice have this type 1 polarized phenotype. And one of the genes, so we wanted to confirm this relatively new technique with a more established approach, but also one that looks at protein rather than this is looking at RNA expression. So we used flow cytometry for just one of these genes, specifically CD274, which is the PDL1 receptor. And so shown here is by flow cytometry. In red is the level of CD274 in eosinophils in the control mice, and then in blue in eosinophils in the myocarditis mice. And you see that a subset of them do express CD274 only in the myocarditis mice, and that it has been quantified over here on the right-hand side. So this provided additional evidence, again, with an established technique and on a protein level, that eosinophils in the heart have a type 1 polarized phenotype. And so in summary for the study that we have done, we concluded that eosinophils are required for heart damage in hypereosinophilia-associated heart disease, and that eosinophils in the heart of hypereosinophilia-associated heart disease are type 1 activated. And so from that and the work that we're working on now, we're hypothesizing that it is these specific subtype of type 1 activated eosinophils that are the key contributors to disease pathogenesis. And the reason I think this is important, we nowadays have ways of depleting eosinophils, but that depletes all eosinophils. And what I think is important is, as we understand how to tell apart eosinophils that are maybe providing homeostatic roles or are at least not being damaging from the ones that are pathogenic, like the bad guys. So if we can tell the difference between the bad eosinophils and the at least not bad or possibly good eosinophils, if we can find ways to target specifically the bad guys, that would be a much cleaner way to try to deplete them and use them in treating disease. And so, if I think I still have, yeah, I still have a little bit of time. I just wanted to spend the last few minutes talking about a project that I don't really have data on, but I'm very excited about that we're working on in the lab, which relates to the biomarkers and the idea of early diagnosis. So, as I've mentioned, the diagnosis of hypereosinophilic syndrome depends on demonstrating, first of all, that eosinophils are not just increased, but that they are causing tissue damage. And then the other thing that's important is knowing which organ is damaged. So, as I've mentioned, if there's heart damage, those patients will undergo much more aggressive treatment and follow up, and different treatment potentially, and different management, different follow up than patients who don't have heart involvement. And so at the current time, troponin is used as the main screening tool, and then cardiac MRI can be used in certain situations, especially if troponin indicates that there is heart damage. But troponin is not specific. Troponin will be elevated, obviously, in anyone who's having a myocardial infarction, so a heart attack, but it will also be increased in a variety of other reasons where there's heart damage, not necessarily related to eosinophils. And cardiac MRI is not something that's used routinely as a screening tool, but because of its expense and availability, so it's only used after we have a suspicion for a heart involvement. And so I think it's really important that we develop biomarkers that will tell us not just that there is tissue damage, but then which organs or tissues are involved. And I've mentioned that one of the things that we did in our mice was to look at those cell-free DNA. And I said at the time, well, the problem with cell-free DNA is that we don't know where it came from, right? We're just measuring total cell-free DNA that's circulating in the blood, but we don't know which organ it came from, etc. However, there actually is a way to determine that. So, and I'm just using an example of these two papers that have done it, this kind of use this approach in with COVID infection, but this can be done with GVH, has been done with GVHD and could be done with hypereosinophilic syndrome. But basically, in every organ or cell or tissue, as a cell becomes determined to be an eosinophil or a cardiomyocyte or something, there are changes to its DNA, not to the DNA sequence, but there are what epigenetic changes to the DNA. And some of those epigenetic changes are stable. So if you were an eosinophil, if you are an eosinophil, that DNA in that cell is going to have some stable markers that are going to be different than markers on a cell that's come from the liver or the lung, etc. And so if there is release of from dying cells, whether it's in the heart or the liver or the fat, that cell-free DNA will have these epigenetic markers. And with admittedly very complicated methylation sequencing and bioinformatics, we can tell which organ individual cfDNA came from. So one can imagine, because this is now circulating in the blood, you can draw blood from a patient and get that cfDNA and then perform this methylation analysis that will tell us, oh, in this patient, the cfDNA came from the kidney or the liver or the heart, etc. And so that is something that we are now trying to pioneer in the field of hypereosinophilic syndrome, as well as eosinophilic gastrointestinal disorders, to be able to use this approach and hopefully overcome some of the barriers with biomarkers in HES. So with that, I would just like to quickly thank my lab and pictured here in red are current members of my lab who have Usman and Ima were the primary authors on the study that I've presented. I just realized I never did put a reference to our paper, but that's okay. And then Ben is a research assistant who has helped significantly, and he is also playing an important role with this cfDNA study. And then below are some of the past members of the lab. And I also want to thank my many collaborators, particularly Mark Rothenberg and Amy Cleon, as well as Maria Medvedovich, who helped with the bioinformatic analysis for the cell-free DNA. So with that, I will stop and I would like to open for any questions.
Thank you so much, Dr. Zimmerman, for such a wonderful presentation and helping us all learn more about our heart and the potential for hypereosinophilic conditions. So now for everyone, I see we've already gotten some questions in, but for those of you who have not asked a question yet, please check out the Q&A tool on your screen. Go ahead and click on that. You can type out a question. We're going to go ahead and have a discussion. Alrighty, so the first question that came in, can EGPA cause damage to coronary arteries and subsequent calcifications?
Yeah, so EGPA usually, I will first say that I don't know for sure if EGPA causes coronary damage. It primarily causes, what I'm more familiar with is that it's usually the small vessels and small arteries that it damages, but I cannot tell you for sure that it doesn't cause coronary disease. So I will leave it at that. I'm not 100% sure.
And we've got one follow-up question about EGPA. What type of arrhythmias are seen in EGPA? And again, completely okay. I know today's talk was focused on HES.
Yeah, and I am really not, as I've mentioned, I'm a pathologist, so this is not in my area of expertise. So I'd rather not answer that question, or I'll answer it with I don't know.
Thank you for your honesty. Alrighty. So if someone has HES and they are curious or wondering about potential heart damage, how would they go about finding out?
Talk to your doctor about your concerns. They will perform, in addition to history and physical exam, and often, especially at diagnosis, but then sometimes if there's any suspicion during the management of continuous disease, doctors will usually perform a troponin test in your serum. And then if there's any other suspicions or other reasons, they might do additional imaging. Cardiac MRI is a fairly good test for it. Some will perform echocardiography, and there's other tests that can be done at this time. So usually troponin for screening and then additional imaging can be added if indicated based on totality of the individual patient.
Thank you. So a nice question came in that kind of tags along with that. How often should patients with HES have their heart monitored?
So I will again say, since I don't personally monitor patients, I don't know exactly what is done in real life. In studies, there are usually, specific follow-ups at 6 months. So again, at diagnosis and then potentially at 6 months. But these would be, I would refer you to the patient, to the doctor that monitors your disease. um, to best answer those questions.
Thank you. Other people are curious about who with HUS is more at risk for heart complications. For example, those with certain levels of eosinophils or with other
Mhm.
coexisting conditions. Like, is it something that everyone with HES should be monitoring their heart, or there's certain people and certain coexisting flags that uh, warrant monitoring?
Yeah, so um, certainly the highest risk appears to be in patients um, with neoplastic hypereosinophilic syndrome. So your myeloid hypereosinophilic syndrome, the ones associated with the translocations and molecular changes that are so when there's a clonal hypereosinophilia, where really it's kind of like a leukemia basically. Those have the highest risk. But it can happen in other patients as well. So um, I think everybody should, at least at diagnosis, it should be considered. And then if anything happens along the way later on, it can trigger additional testing. The level of eosinophils, I don't think we have really a good cutoff. I mean, we usually consider eosinophilia over 500 and then hypereosinophilia over 1500, but that's not just for the heart. That's sort of a general cutoff.
Thank you. How often are you seeing heart problems in HES patients?
Um, so based on the studies, and again, I don't have my own personal cohort, um, it's about uh, 1 in 5 if you take all of HES will at some point in their disease have some problems with the heart.
You've talked at different points in time about the numbers of eosinophils. We've got a follow-up question here about what is an alarming number of eosinophils on
Mhm.
blood work?
So I would say anytime it's over 1500, we would certainly warrant um, a quick workup. But if it's persistently over 500, we do would also perform a workup and, try to understand if is there a explanation for it, because sometimes, patients with EGID or asthma can have eosinophils in the 600, 700, etc. So if you know that that's what's going on, um, you might not be as alarmed. But in somebody who doesn't have any of those underlying conditions, I would say anything above 500 should make us think what's going on. But then once you're over 1500, then we know that something uh, definitely is going on and we need to figure out what it is.
And some questions about some biologic treatments as well. Are you comfortable
Mm.
talking about mepolizumab and the potential use cases for that?
I mean, I don't really, I probably not. What I know is from research studies. I don't particularly personally treat patients, so I would not be the best to answer those questions.
Maybe let me scan through some of these questions here. Alrighty. Did your research compare different treatment strategies to
Mm.
see which is best for heart patients, or is that something you might look at in the future?
Yeah, that's an excellent question. Excuse me. It's certainly something that we we as in the community need to get into. So no, we don't yet have that type of comparative studies, but it's certainly something that should be done and is important to do. And
Thank you.
can I just get back to so the in terms of plans? Um, I mean, there is a fairly strong eosinophilic community that is trying to. I've shown you that one study where there were multiple institutions came together and were trying to combine their data. I really enjoy working with my colleagues from around the country and around the world. There's European collaborators. It's a very strong community that tries to work together and not compete, which sometimes happens in research environments, but really we recognize that none of us can do anything alone, considering how rare this disease is, and that uh, it's best if we work together. So this community does work very hard to try to answer some of these questions.
Well, thank you. As you think again about kind of the future of your research and the work that you're doing, how might it inform both future treatments and diagnostic tools?
Yeah, so I think that um, to me, understanding the mechanism of disease is really critical to us coming up with ideas of how to diagnose and then how to treat. And that's part of why I wanted to show you the cell-free DNA, because that idea really came from our mouse study. And then now we are actually working on a biomarker. And then what I didn't talk about is this receptor, CD274, the PD-L1 receptor, that has been considered, that's there is a drug that's approved for CD274 in cancer. So could this be something that we could repurpose potentially if it shows to be important in pathogenic eosinophils? Could this be something that we could use here? So I think, and we have a lot of work to do to better understand some of the questions. Again, I'm really fascinated by why do some patients get heart disease, some get a skin rash, some get nothing, even though they have a lot of eosinophils. So understanding that and then understanding how these eosinophils do the damage to organs, I think will be really critical for us to be able to narrow our treatment and come up with new ideas for treatment as well.
Thank you for all the information that you shared today and for all the research that you and your team are doing. We appreciate all the contributions to be able to help patients with HES and other eosinophilic conditions. Thank you also to our education partners for their support for this webinar series, including AstraZeneca, Bristol Myers Squibb, GSK, Sanofi, Regeneron, and Takeda. Our next webinar will be on December 9th. We hope you will join us to learn about preparing your child for healthcare independence. For this webinar, we will be joined by Dr. Nicholas Kajacob and Dr. James Calloway, both from the University of Alabama at Birmingham. We've got one as a pediatrician and one as an adult provider to be able to share kind of more information about that transition. To register, you can go to apfed.org/webinars. You can also continue learning more about eosinophil-associated diseases by subscribing and listening to our podcast, Real Talk, Eosinophilic Diseases. In the latest episode, we talked with Dr. Andrew Lee about TSLP and EOE. It was an interesting discussion about a potential treatment target. You can find the episode at apfed.org/podcast, as well as on Apple Podcasts, Spotify, Audible, and more. We're also excited to get everyone to start marking your calendars and saving the date for our annual patient education conference. So for 2026, we are headed to Denver, Colorado next July. You can see more information as we release it at apfed.org/conference. There's also still time to continue watching the conference recordings from this summer on demand through the end of the year. Finally, we encourage everyone to continue the discussion started today on our Inspire network. It's a great place to connect with patients and caregivers for support. You can check it out at apfed.org/connections. Thank you again for joining us today, Dr. Zimmerman, and for everyone who joined us for APFED's EOS Support webinar series. If you have a question that didn't get answered, please email us at mail@apfed.org. I hope everyone has a great afternoon.